Hello and welcome to the Beyond Biotech podcast for August the 19th. And it's podcast number 10. Double figures already. Just another 90 to the 100 mark then. 100 mark actually sounds like an old German banknote, and there's probably a whole generation that have no clue what marks, francs and pesetas are. I'm Jim Cornell from Labiotech, and we're back to 3 interviews on the podcast this week. More on that in a little while. And it was a bit of an up and down week, a couple of days with news coming in like a torrent and a couple where it was more like a trickle. And it seems like a decent analogy, as there are some parts of the world putting up with lots of rain and others suffering from a drought. Today is National Potato Day, and if I'd been really smart, I would have done an interview with a biotech company using potatoes for something innovative, but I didn't. This week, thanks to the nice weather, I did manage to get a nice walk in around a loch, otherwise known as a lake, but it was more than a bit disappointing to see all the trash left by campers. You'd think that if they went to all the trouble to go out and enjoy nature, that they'd appreciate it enough to not mess it up. Today's also the last day of school holidays here in Scotland, so the TV will get a nice rest, and I'll get back to that pile of new music that I haven't listened to yet. But I must say I do enjoy the company. No doubt within a week of going back there'll be a day off somehow. I'm not sure when the next public holiday is for us. It's a bit odd with my co-workers in so many different countries, in Germany, France, England and Spain, so if we all took off all of the holidays in those countries, we'd probably never get anything done. But it's good to learn about the different holidays and reasons behind them. Alright, I should tell you who is on this week's episode, and so I will. Our guests this week are Endpoint Health CEO Jason Springs, Xianghao Zuo, Deputy Director of R&D at Triastek, and Andy Whiting, CEO of Nevrargenics. And I guess that brings us to the news. As I mentioned earlier, it was busy, then not, then busy again, but the great thing about news is that it's unpredictable. We do seem to get these 3 waves of information throughout the day. We get the Asian companies to wake up to, and then the European press releases, and in the early afternoon, it's the North American news that starts to roll in. Or sometimes it doesn't. So, here's some of the news you may have missed over the past 7 days at Labiotech.eu. The FDA lifted a hold on a lymphoma study, 89Bio completed enrollment in a NASH trial. A collaboration called the Project to Accelerate New Treatments for Tuberculosis was announced, and TurtleTree has produced the first precision fermentation sustainable lactoferrin. A Scottish biotech startup has received funding to create sustainable omega-3. We had an in-depth article on the challenges of developing radiopharmaceuticals for cancer, and researchers have been invited to apply for DNA synthesized by Evonetics platform. Viridian reported positive news from an ongoing trial for patients with thyroid eye disease. Programmable medicines company Senda Biosciences closed a $123 million Series C financing, and Merck is investing up to $37 billion into an Orna Therapeutics circular RNA collaboration. We had an article on biotech companies in Asia. The Aptinyx fibromyalgia drug missed its endpoint. Roche announced approval of its influenza drug and a new COVID test, and Shortwave Pharma is starting a clinical trial for anorexia nervosa treatment. Scientists have developed a gel from spider silk proteins that will have biomedical applications. Small Pharma's major depressive disorder clinical trial was approved, and we had a look at 5 of the hottest biotech companies in one of my favourite cities, San Diego. And you can read all of these, and at least as many more, at Labiotech.eu. And that brings us to the first of this week's guests. Nevrargenics is a UK drug discovery and development company focused on innovative and novel medicines for neurodegenerative disease. And this is another interview that was done by our own Liza Laws.
I'm Andy Whiting. I'm the CEO of Nevrargenics Limited, founder of the company as well, together with colleagues that we've been working together with for a lot of years, but we founded the company towards the end of 2019 to exploit results in the area of developing small molecule drugs to treat neurodegeneration. It sort of stems from a project which has its history a lot further back in time than that, but we were getting to the stage where we had some really interesting IP developed within Durham University and Aberdeen University, so we thought we'd spin it out as a company. The reason for doing so is twofold, really. Firstly, as I say, that we had some, perhaps to a certain extent, unexpected results. We weren't intending to look to develop drugs to treat neurodegeneration. We were trying to understand the mechanisms of neurodegeneration so that perhaps others might be able to solve the huge met need that is there. I think since I've been CEO of this company, we've been looking to raise money to finance the drug development project forward. What's been fascinating and sad in many ways, I suppose, is that everyone we've talked to has been touched by neurodegeneration, either directly through partners and immediate family or close friends. And that even includes myself. So here we are sort of creeping out of COVID across the globe. And we see that as a pandemic. But neurodegeneration is almost the silent pandemic. It is vast, and the struggle to deal with it appropriately is just huge. And a lot of that stems from the fact we're only just beginning to really understand a lot of the causes and often linked underlying causes between all of the different neurodegenerative diseases. That includes Alzheimer's, Parkinson's, MS, ALS, FTD, schizophrenia, all of these types of neurodegenerative diseases that, amount to pretty much around 1 billion sufferers on the planet, so it is a huge problem. So really our key interest is to look to try and tackle that. And that's what Nevrargenics is all about, really.
Could you explain where some of the first ideas came from, particularly the relevance of vitamin A, which comes from things such as Brussels sprouts? If you could just explain sort of the initial scientific ideas.
Yes, I mean, that's we've slightly tongue-in-cheek written articles about Brussels sprouts not being just for Christmas, the old thing about a dog not being just for Christmas. Well, the good thing about eating brassicas and, your sprouts at Christmas and things of that kind is that you've got lots of carotenoids, lots of these sort of orangey pigments in them, which are a major source of vitamin A in our diet. So when you eat those good greens and together with other kinds of foods as well, you can metabolize these carotenoids to vitamin A. And vitamin A, obviously, many people take it in supplement tablets and things. It's one of the many essential vitamins. It's shuttled around the body to all different parts of the body. All mammalian cells require vitamin A, and it's used not only in its own right for various things, but it's oxidized within cells to retinoic acid. It's also oxidized to a sort of an intermediate oxidation state known as retinal. And some of your listeners may obviously understand its key role in vision. It's used as a substrate for detecting light, obviously, in the eye. So these metabolites, these different analogues of vitamin A that are produced in all sorts of different cell types and tissues, are absolutely essential, and they control cellular development and growth and everything from life and death and everything in between, really. One of the key things that retinoic acid does, particularly in neural tissues, which of course is the relevance really to what we're talking about here, is that certainly in early stage of embryonal development, have a key role on the development of the CNS, of the central nervous system, of the neural system, the differentiation, so the transformation of stem cells into neural type tissues. And that essential role carries on throughout life. And retinoic acids even have roles in switching on and off apoptosis and other types of cellular health. So they're a key known as a signaling molecule. So they interact with nuclear cellular receptors and trigger these different events of the different life stages, if you like, of the cell development. So these are the really important compounds. About 15 years ago, we came up with the idea of making synthetic analogues of these. And this would seem a slightly odd thing to do, perhaps, given how, in fact, it's not particularly difficult to make retinoic acid, and it's made on large scale. Vitamin A is made on large scale. I think many people have skin creams with vitamin A in them, with retinol in them. So they probably know that it's easily made. But the trouble is the natural compounds are really quite unstable, both to light and air. And actually, that's why they're made in situ within cells from vitamin A. So we were wanting to make synthetic analogues just to see what they did in order to understand mechanism, because once vitamin A is oxidized to retinoic acid, it's also isomerized into a number of different isomers. And in fact, there are quite a number of other metabolites which are oxidized analogues as well. And we think there are somewhere in the 20s of compounds of that type made in greater or smaller quantities depending upon on their role. And the main one is one known as all-trans-retinoic acid. It's the compound that's most understood, most studied. It is used to treat some diseases in its own right. So we wanted to make, it was a curiosity-driven research project, wanted to make analogues of these things and just see what they did, because we didn't have a proper understanding of their role in a crude way, yes, but we didn't know what individual stereoisomers, individual molecules did, what was their particular role in different cell types. And so we made some of these synthetic analogues to mimic the natural ones just to see what they did within biology. And we found some weird things and some really interesting things. And that eventually really led us to, I guess, kind of put together a bit of a manual on how to design drugs potentially, because we started to understand some of the key downstream effects that those structural changes would have on the biology of these compounds at the molecular level. So that allows you to think, okay, how can you turn what's a molecular probe into something that's potentially a drug? And so that's really how we came at this.
With many things, we kind of know, we being sort of, the general public generally know if we're depleted in something. There's been big things about vitamin D and, we can go and buy supplements. Is there any way of knowing that you would be depleted in vitamin A? Are there any sort of common signs?
That's an interesting question. There are, I think you would be generally really quite unwell. The problem with retinoic acids and vitamin A analogues in general is they're involved in all sorts of things. I think the earliest stages are probably eye deficits and a reduction in eye sensitivity. I mean, that's one of the reasons that you're often told, eat your carrots because it's good for your eyesight, helps you see in the dark. Yeah. And that's quite literally true. So I think some of the early indicators, but you have huge numbers of symptoms because of their key role in so many pathways. The bigger problem often, sometimes people, of course, over self-medicate by having supplements. And there are examples of people having too much, for example, carrot extract and things of that kind, being a bit too obsessed. I mean, if you have a healthy diet, with vitamin A sources and including plenty of green matter and anti-carrots and things of that kind and sprouts and things, there isn't really a problem. But some people who've got obsessed with it actually have the opposite problem of getting the more sort of retinosis type characteristics from over-toxicity, because vitamin A in large quantities really quite toxic. It's stored in the liver.
Retinoic acid receptor modulators, they're key to your work, but what's their role in neurodegenerative pathogenesis, and how are vitamin A metabolites key to cellular health?
Retinoic acids in general modulate and tune cellular activity in all tissue types to different extents, depending upon what cell or tissue type you're talking about. When it comes to neural health for adults, one of the key things that retinoic acids do is that they particularly interact with retinoic acid receptors. So the way in which these signaling pathways work is that these small molecules, where vitamin A is concerned anyway, initially, the natural process is vitamin A shuttled around by a large sort of protein carrier. It's carried to individual cell types. The vitamin A is then carried across the cell membrane, and then carrier proteins carry it across further, different carrier proteins, once oxidized into retinoic acid. So you have a whole raft of machinery, enzyme machinery, a couple of oxidation reactions. The retinoic acid interacts with these so-called retinoic acid receptors. Now, these membrane receptors, they're based around the nuclear envelope. And what they do is that once bound, they become activated. And then there's a bit of a cascade of processes where they interact with other nuclear receptors and chaperone proteins, all sorts of things. But basically, they bind a section of DNA and they cause transcription. So that could either be an upregulation process to switch a particular gene on, or it could be downregulation to switch a particular gene off. And those might be genes to do with all sorts of things. But where we're talking about neurodegeneration is particularly inflammatory pathways. So one of the things that we know in sick neural cells is that inflammatory processes, often as a result of oxidative stress, start to become problematic. And retinoic acid, the right interacting with particular receptors, there are quite a number of these nuclear receptors. We're interested in these so-called RAR retinoic acid receptors. There are other types, RXRs, the retinoid X receptors, and there are even RORs as well. So, I mean, there's a number of retinoid-based receptors, but we think it's particularly the retinoic acid receptors, the RARs, that need to be modulated. And what you can do by hitting the right combination of those receptors is switch off certain inflammatory pathways. And you can also switch on anti-inflammatory pathways, which help give the cell positive response to dealing with oxidative stress. Now, the first impact that can have is to slow down neurodegeneration, because as a result of oxidative stress, cells produce pathogenic proteins, and some of those proteins aggregate into an even more toxic form. And in different neurodegenerative diseases, you get different types of these proteinopathies, so these toxic proteins that can build up and aggregate. Now, although some of these are certainly toxic in their own right, of course, the damage is already done. So they're symptomatic of the fact that cells are under stress and already beginning to suffer problems. So by adding external retinoic acids, what you can do is you can stimulate the upregulation of these retinoic acid receptors in their own right. So one of the things that you see in Alzheimer's and other diseases is you see a downregulation or a loss of these key signaling proteins. Now that limits the cell to respond to stress. So the more stressed it gets, the more sick it gets, the less retinoic acid receptors there are, the less it's able to deal with the stress. You can imagine it's a vicious cycle. So what we're thinking about really is turning that vicious cycle around into a positive one. So you add exogenous compound, it mimics a particular retinoic acid, it stimulates further retinoic acid receptor production and starts to reduce stress and inflammatory effects, and at least hopefully stop the disease. But of course, if we get this right, the potential is to reverse this kind of stress-based damage to these cell types. So we're also potentially looking at a neuroregenerative type of process as well. And certainly in vitro models show us that might be possible. We can trigger neurite outgrowth, for example, in cell types, and that could be a model for these types of cells. Sort of neurite outgrowth can start interlinking with each other, and that's a model for regeneration or so-called improved neuroplasticity. So the ability of these neural cells to rewire themselves and regain function. So we're not talking about a single pathway. And what I'm talking about there, these retinoic acid receptors are mostly in the nuclear envelope, but they are also expressed in the cytoplasm. So they do control other processes. For example, some phosphorylation reactions are also controlled by these things. So when you design drugs to hit these receptors, what you want is to hit the right ones, not the wrong ones, upregulate the good processes, downregulate the bad processes. And we've made compounds that hit the wrong receptors as well as hitting things that hit the right receptors. So we're getting to understand incredible subtlety. And these are compounds that we think of as multimodal in that they aren't just hitting one particular target. They may be hitting more than one, but it's the balance of selectivity between the different targets and the balance of the downstream effects that they have, depending on where those targets are. So whether in the nucleus or in the cytoplasm. So it's a far from simple picture, to say the least. But I think one of the things that we're particularly interested in, and of course we hope to develop a drug that's effective for the huge number of people that suffer from these awful diseases. But actually, even if we weren't successful, if we can develop information that helps the next generation of medicinal chemists to design better drugs than we're able to do if we're not successful, we would be incredibly happy, because clearly the goal is to solve and understand neurodegeneration in all its forms. And it's not simply about getting a drug on the market. There's a bigger picture here, obviously, and a huge goal to try and really better understand along the way what is going on and what you can do to stop it.
Where exactly are you at the moment, and what's your next goal / milestone?
We're just a little bit over halfway through our first major investment round. We're sort of writing big grant applications, EU, Innovate UK type things to bridge finance. In terms of the practicalities of what that delivers, we're sort of intermediate preclinical. We're just going through efficacy studies. We have a lot of in vitro models that show phenomenal activity, and our drug is phenomenally potent. So we're looking at often nanomolar, subnanomolar levels of exposure in models and cell systems, and seeing huge impacts on the cell models that we're looking at. We're translating that across into in vivo models, into animal models. We did a very short 6-hydroxy dopamine rat-based model with one of our collaborators, Professor Susan Duty down at King's. That's now being extended to run for a number of months. That showed some phenomenal results. And we've just signed off agreements and paid some deposits towards ALS-based models, an MS-based model. So we're in the efficacy stages to substantiate our view that these should be valuable in a number of different diseases. And that allows us to do two things. One of the key things, obviously, is that it gives us evidence that a particular disease is worth targeting because we see effect in those animal models. So the ALS-based model, initially an SOD1 model that we're looking at, we'll do some other ones if successful as well, will allow us to look at tackling ALS. I think I mentioned that a little earlier, which is heavily linked to FTD. And for FTD, frontotemporal dementia is often seen as the head end of ALS, as well as one of the motor neuron diseases. So they're very much related. And that's where we have a strategy, an orphan drug development strategy in that disease area. It's an area of huge unmet need, not vast numbers of patients, but it's not about making money. Although I think it will be certainly commercially viable. It has to be, because otherwise we don't make enough money to develop the next drug for the next disease. However, it's probably the disease of greatest unmet need, so that's where we're kicking off. It's a horrendous disease. Typical lifespans are between 3 and 5 years at best, and we want to substantially change that. And of course, because these people are very massively debilitated very early on, as I say, it really is an awful disease to observe. So I think that's where we're kicking off for a number of reasons. But of course, the other side of it is if we can demonstrate efficacy in a number of different disease models, it provides evidence and comfort, we hope, especially when we talk to bigger investors at the next stage. So what we'll need at the next stage, drug development is unbelievably expensive, and we will need, ever increasing amounts of money for developing the next stages. So of course, if we can demonstrate to potential investors that we have a platform-based technology, it can apply to different diseases, the same types of drugs, or tune the structure of those drugs a little bit to match different diseases, it's something that we want to try and do. So I think it's a two-pronged thing about improving our chances of better investment in future, but it's also very much about proving the viability of our drug development programs. So that's what we're using current investors' money for, and we're nicely underway. We will have a lot more evidence of where we are in effect, actually, by the end of this calendar year. So those efficacy studies will be reporting in roughly that time scale. Beginning of next year, then we're planning to make plans, complete the plans, assuming good efficacy in these models, to do the translational work in the NHP systems. And then hopefully, it might be a very tight time scale, but we're aiming around 2024 to get into patients. It's very much a very tight time scale, but that's the program that we're on.
Triastek has developed 3D printing technology to provide solutions for drug development. It recently signed an agreement with Lilly to focus on improving the bioavailability of drugs in the intestine. To tell us about that and the company is Xianghao Zuo, the company's Deputy Director of Research and Development.
All right, so I guess to start, I wonder if you could tell me a little bit about the company first.
Of course. Triastek is one of the leading 3D printing pharmaceutical companies and the leader in metal extrusion technology for pharmaceutical product development and manufacturing. We offer end-to-end solutions encompassing novel dosage form design, development, and continuous manufacturing at commercial scale with flexible product capability. We created the pharmaceutical formulation by design platform using digital platforms for the formulation development and continuous manufacturing.
And so the actual 3D printing, could you tell me a little bit about how that actually works?
Yes, 3D printing pharmaceutical technology is a foundation for a wide range of applications in pharmaceutical areas, such as new drug development and development for 505(b)(2) products. It is an innovational manufacturing technology. Detailing about MED technology, it is a continuous technology that continuously convert the powder materials into the molten state, then extruded and precisely deposited on the plate to form the structure layer by layer. It has no filament preparation needed. We will have a digital design and the processing for both R&D and high throughput manufacturing. We will have digital structure design. We will have a digital print code generation, and we use SCADA system for the QC online, and thus we got 100% QC. And for the development of the formulation, we developed a formulation by design. So we start with a PK profile, and we derive this PK profile into our in vitro release profile. Then we will be able to design the tablet structure using software, and we will select the materials from our database. Then we will perform this in vitro dissolution validation. And if it meets our requirements and it meets the demand, we will perform in vivo PK profile. And if we find there's a little vibration, we will calibrate with a very high efficiency. We think the advantages of MED technology is it doesn't rely on the filament and it doesn't use any solvent, so there's no post-printing process, which is a good benefit for the environment. And we can also lower the processing temperature for better stability of the API and the excipients. And also we will have like high printing precision.
And are there any other benefits to 3D printing for drugs over a traditional method of creating drugs?
Yes, we think the first thing is that 3D printing can do faster prototyping and also require a small amount of API. According to the ebook published early this year by our partner, tablet printing for clinical trial supply has the potential to reduce required API by 50% using 3D printing technology. And the development time has the potential to be reduced by 60%. The second thing we think is the flexible and complex structure design. And we can provide solutions for challenging problems such as column targeted drug delivery. We can also do pulsatile drug delivery and gastroretentive drug delivery. And another thing is continuous manufacturing. By continuous manufacturing, we can improve the safety and quality of the products. And as I mentioned before, we use SCADA for the online monitoring, so we can get 100% QC. The other thing we think is flexible production capacity. We combine printing units to generate a high throughput production. So our printing units can be combined at will to meet the demand of the production. The last thing, but not the least, we think we can do personalized dosing. So we can use the same material composition, and the dose can be adjusted easily, and can also produce tablets with precision combination of different APIs.
And one of the biggest issues, I guess, with any drug, however it's created, is the drug release. So how is the programmed drug release achieved?
We use formulation by design to during our product development, and we can do in different methods. For example, we can have single API with different release profiles. We can have one API with immediate release and the same API with extended release. So we can have a simulation of both the immediate release and extended release at different ratios. So we can get different release profiles as we design. And we also can do development of multiple APIs with different release modes or rate in one tablet. And by combining multiple APIs with different release kinetics, complex dosing regime can be achieved to meet the different clinical needs. For example, if we have like a multi-compartment design, and we can have each API to be printed in one compartment, so no chemical compatibility issue will happen between different APIs. And we can also have the APIs in compartment and control the thickness of the delay release layer. So we can adjust the release profile to meet the requirements of onset time. And we can also do structure and the surface area of each compartment to change the surface area and the structure. Thus, we can control the release rate. So in this way, we can greatly improve the patient adherence and when the combination therapy and program release of multiple APIs desired.
How did the partnership with Lilly come about?
We first built the connection at the end of 2020. And since then, Triastek and Eli Lilly has in-depth communication on the principles of MED 3D printing pharmaceuticals and application in formulation development. We think fast prototyping and the flexible structure design to achieve program drug release profile are the advantages of 3D printing technology. So the collaboration project aims to use 3D printing technology to precisely deliver drugs to target sites in the gut at a very high efficiency. So in the collaboration, we will take a deep dive into excipient properties and process parameters to conduct an in-depth study of how these factors impact drug stability. And then we will identify unique 3D printable structure design to deliver drugs into targeted parts of the intestines and achieve program release profiles, and to improve the bioavailability of the 3D printed drugs when administered orally.
And is this something that would be applicable in a lot of different drugs? You can tailor this according to the different specifications?
Yes, we think so. We think MED 3D printing technology can be applied to all BCS classes. For example, for water-soluble APIs, MED can print the small particle dispersion with the API in crystalline form. And for the low solubility APIs, MED can print the molecular dispersion with the API in amorphous form. We are also making efforts in peptides and soft gels.
And do you have any other partnerships in place other than with Lilly?
Yes, sure. We have a lot of other partnerships right now with our product pipeline in different stages, and we have novel platform. We have different kinds of collaboration. For example, we have co-development with domestic companies and for like rare disease and 505(b)(2) disease. And we also do feasibility study to provide novel design or do faster prototyping with the domestic companies and the other MNCs. So besides Lilly, we have joint projects with Merck, and we also do co-webinar and co-marketing with them. And we are also in discussion with other MNCs.
And I guess recently you had clearance from the FDA for your other printed drugs, T19 and T20. I wonder if you could tell me a bit about both of those.
I will be very glad to introduce our T19 and T20 products. I will start with T19. T19 is our first product, and it's our second 3D printed drug product registered with the US FDA. It is used for rheumatoid arthritis. It has huge clinical and market demand. So our tablet design was based on the principles of chronotherapy to fit the circadian rhythm. So when the patients take T19 before going to bed, the drug concentration in the blood reaches the peak in the morning when the symptoms of the disease such as pain, joint stiffness, and dysfunction are the most serious, and maintains its blood drug concentration during the day so as to obtain the best drug treatment effect. This product has received both the FDA and NMPA IND clearance. We are now carrying out a pilot PK study, and our next step will be pivotal PK study. And I will also introduce our product T20. T20 is our second product. It's the world's third 3D printed drug product received IND clearance from the FDA. It is a BCS class 4 drug with low solubility and low permeability. It is used for the treatment of cardiovascular and clotting disorders. Current product was taken twice a day. So we use a 3D structure design to provide once daily dosing while being anticipated to maintain the same efficacy and adverse effect profile. And we will also improve the patient adherence, which is the most important. We fine-tuned the PK/PD profile with the multi-compartment design for this product, and to allow right amount of drug to be released at the right time and right GI tract. So different amount of API was printed in each compartment and delivered to different GI sites. And the release onset time and the release rate were also precisely controlled with the structure design. This product has received the FDA IND clearance early this year. We're now carrying out our first pilot PK study.
All right, that's excellent. Those are the only questions that I had. Is there anything else that you wanted to cover?
I think I can make a little bit of this MED 3D printing technology for the industrial manufacturing. Our MED printer nodal array can be designed. So we design a single nodal for like R&D printer, and we design nodal array for the continuous manufacturing. And the nodal array design enables mass production using multiple materials to fabricate complex structure. And for the product line, it's also a continuous manufacturing product line. We use powders of the APIs and the excipients at the beginning, and we got the packed product at the end. Each of the product we achieved like 100% QC by using processing analytical technologies and SCADA to monitor the printing process. And we use these methods to acquire data. And so the data are traceable for each tablet, and we will have a QR code for each tablet. So the data of each tablet can be read in the future. As I mentioned, we also have like flexible production capacity. We can easily change the combination of our printing units. And to sum up, we think MED technology offers an end-to-end solution encompassing novel dosage form design and formulation by design, product development, and the continuous manufacturing. And we also, with the design of tablet structures, we can also do like the delivery of low solubility APIs where sustain supersaturation. And as I mentioned before, we can do program the release of APIs at a targeted location in the GI tract with the right rate and the right dose. And the MED technology can also offer a means of formulate challenge compounds to accumulate the biopharmaceutical efficiency where like gastro retention enhancement of auto absorption of biologics. And we are also making efforts on like children's medicine. We're trying to print like hydrogels, which will improve the adherence for the children.
Now we head stateside to Endpoint Health, a therapeutics company dedicated to addressing unmet needs in immune-mediated acute and chronic diseases. It recently received $52 million in equity and debt financing, and to give us the whole story is Endpoint Health CEO Jason Springs.
We're a precision immunology company, and what we do is develop precision therapies. So that's new therapeutics paired with companion diagnostics, really designed to pick out the right patient based on their unique biology that we can see with our technology. And the intent is really to kind of bring the benefits that we've seen in other clinical areas like oncology, bring those benefits of precision medicine to a completely new set of patients suffering from diseases driven by dysregulation of the immune system. And that ranges all the way from areas like autoimmune disease, things like inflammatory bowel disease, systemic sclerosis, rheumatoid arthritis, even all the way over to very, very deadly and critical illnesses like sepsis, acute respiratory distress syndrome, which actually are the high mortality causes that you get from things like COVID-19 and other infectious diseases. So really a broad mandate, bringing precision to a new area.
And I guess one of the most recent announcements that you had was a $52 million funding, which is partly for the Precision First platform. Could you explain what that is and what the funding means?
Yeah. One of the things that makes our company unique is what we call our Precision First platform. So the way that we actually develop new therapies is by starting with large sets of patient data and really digging in and figuring out what's going on with the underlying patient's biology, with their immune response in these immunology indications. And the way that platform works is we start mostly with gene expression data, and we built a really unique and powerful machine learning technique that allows us to look at this patient data and look for unique patterns of underlying biology that we believe, and what our data shows us, would lead patients with different signatures to respond differently to the same therapy. And we've seen that pattern in other areas of medicine where patients with maybe a particular mutation in their DNA may respond preferentially to a particular therapy. Here, since we're dealing with the immune system, what we're looking at is patterns in immune response that are dynamic. The output of this whole process is, really two things. It is understanding what therapies or what targets to hit that may be most ideal for a particular patient set in order to find, higher levels of response to a therapy. And the other thing that comes out of the end of this is the ability to develop companion diagnostics, because understanding data, understanding large sets of patients and how they may break into different subgroups is great from a technical or academic perspective. But at the end of the day, you have to build the ability to know what therapy to give to a particular group of patients. And you have to be able to actually find those patients, with a rapid test, for example, a blood test in clinical trials. You have to be able to see who should get a therapy. And of course, when you fast forward to the future, if you have approved precision medicines, you need an approved test, a companion diagnostic to actually find them. So that whole process of starting with patient data, using proprietary machine learning techniques to understand where there are different subgroups of patients, and developing therapies for each subgroup along with a test to find those patients that may respond is what we call our Precision First platform. And you're right, we did recently announce a $52 million financing round. We're very proud of that, especially in the current market conditions, although I'm maybe a bit bullish on the recovery. And that financing is meant to do a number of different things. One of them is to expand our platform, and that means expanding the data access that we have. We run our own observational trials, capturing patient data, and also access other proprietary and private data sources. But it also is dedicated to progressing our pipeline of therapeutics. We've publicly announced we have a partnership with Grifols. We used our platform to identify antithrombin 3, a novel blood product used for anticoagulation. We think that therapy has a unique chance to really resolve some high morbidity, high mortality patient problems that we can spot in a subgroup of patients with septic shock. We also are looking at expanding our therapeutic pipeline into developing novel molecules more in the autoimmune and chronic inflammatory illness section of the market. And we'll be moving more towards that as we progress through the rest of this year.
I guess you mentioned septic shock, and it seems to be kind of in the news quite a lot lately because of COVID, and monkeypox is the latest one with secondary sepsis. I guess you're working on that. Could you tell me about some of the things that you're doing in that area? Because I guess it's a bit muddy in terms of some people can be harmed by treatments and others are helped.
Yeah, traditionally, that's been a tough topic to understand. I think if you back up and think about what happens with a patient's body when you get an infection, whether you look at influenza, whether you look at COVID-19, monkeypox, or really any infection, one of the big medical challenges that can happen is when you get an infection, if your immune system starts to, let's say, go off the rails, when you have a dysregulated immune response, you're having too much inflammation, you're having cytokine storm, you're having immune suppression, all these pro and anti-inflammatory processes can go way, way out of control. That dysregulation, your overbearing immune response to an infection, is what is typically called sepsis. So that's really the clinical definition. And the big challenge of sepsis, whether you have shock along with it, which is what septic shock is that it's very heterogeneous. Not everyone gets it. And when you get it, not every patient is really the same. It is a medical syndrome. The way the clinicians see it today, they can just tell that there's a runaway reaction with the immune system, and it leads to very high levels of mortality. And just to put it in perspective, you have as many, if not more, people dying from sepsis globally from many, many different infectious causes. You have as many people dying globally from sepsis as die from cancer each year. So it's probably the world's most unmet medical need. One of the things that's interesting within an area like sepsis is that you have coagulation challenges, you have inflammation, and you also have immune suppression. So you have this mix of patients where the treatment for one trait, if you give it to the wrong person, it may actually cause harm. If you give an anticoagulant to someone that does not have blood clots, you may cause bleeding. If you give an immune suppressant to someone whose immune system is trying to successfully fight off an infection, you could cause some problems. And so this is really a classic case of where a precision approach, being able to see exactly which patients need an anti-inflammatory because they have too much inflammation and treat them specifically is important. Being able to see which patients really are having a dysregulation in how their blood clots or does not is critically important if you want to try and resolve blood clotting as an issue, which is something that we're really looking at with our antithrombin 3 asset. And of course, there's patients also that have immune suppression where their own immune system really is exhausted. There's just not enough there to fight back whatever virus or bacteria that is attacking their body. And they may need some support with novel therapies as well. And I think sepsis has long been an area where people have very good hypotheses on what therapies should be used, but they've lacked the tools to pick out the right patient. And this is an area where we're really applying our platform and where we've partnered with Grifols to bring in a novel therapy to pick out patients very specifically based on their biology and treat them appropriately or develop therapies with the intent of treating them appropriately.
And I guess you're also doing work on acute respiratory distress syndrome. Could you tell me a little bit about that?
Yeah, that's another very complex downstream effect of infections, but also injury. Acute respiratory distress syndrome, I think the quick way to think of it is a more localized, dysregulated immune response in the lungs. And I think it was a term that not many people knew until COVID-19 hit because it is a respiratory virus, and that became a relatively common critical illness that was caused as a result of the viral infection. And there's really a couple of areas where we look at acute respiratory distress syndrome. It occurs about half of patients with ARDS also have sepsis, and also it occurs in the critical illness unit of hospitals. That's where you end up. And so we've also developed a number of techniques to use patient data to really separate patients into multiple subgroups. And what we've seen is that not only might a number of novel therapies preferentially work in one of the subgroups of ARDS patients we see, we've also published on the fact that we can see a number of existing techniques that are currently used to manage these ARDS patients may work better in some patients rather than others, and we can identify which group they may work in. And so there's a number of different therapies that are used. Patients are put on ventilators. There's a number of therapies to keep them very still while they're on ventilators. And we think there's an opportunity there to use patient information both to better manage patients as they exist today and to look at potential novel therapy development. And we're working internally ourselves, but also talking with a number of different therapeutic partners potentially to look at ARDS applications and how we can really bring a precision approach to that patient population as well.
Could you kind of give me a little bit of an overview of what you have in the pipeline and what kind of timelines that you're working with?
Our first program in our pipeline comes from a partnership with Grifols. So we have in-licensed antithrombin 3 for use in a subgroup of sepsis patients that we can identify with our unique technology. Specifically, this is a subgroup of patients we believe have need for a novel anticoagulation therapy, specifically antithrombin 3. This therapy, we're progressing towards submitting an IND later this year with an eye towards moving into a phase 2 trial next year. We believe that we can go directly to phase 2 because this therapy has some unique history. One, it's actually already an approved therapy in the United States for a completely different use, but it has some really great background and safety with it. It's approved for a rare antithrombin 3 hereditary disorder. And also this molecule in particular has some history of development in sepsis. So we believe that with our platform and the particular background of this, we may move directly into a phase 2 trial, which is a kind of a unique part of our business model. We can look at both brand new molecules and take them into humans in a more traditional path, but also identify new clinical paths, new uses for therapies that have already been developed, which gives us an opportunity to make sure we get therapies that are needed by patients into trials, into patients, and into the clinic, into the hands of clinicians a bit faster than a more traditional development approach. We have a couple of unannounced programs, but we're looking at pursuing our second immunology indication, filing an IND early next year for our second program. We haven't announced a particular area yet, but as I mentioned, one of the uses of our funding is to expand our pipeline out. And I would say stay tuned for more information there as we progress through the rest of the year.
Is there anything else that you wanted to talk about that we haven't covered?
Yeah, I think the biggest thing for us, what we're really proud of, if you back up a lot, we spend a lot of time talking about technology and pipeline and therapies. But what makes us very excited really is being able to look at a big area like immunology, which has, I think, had a pretty traditional drug development path, over history. People come up with a target and they see which patients they could develop a particular therapy to hit that target in. And what I'm very proud of, and I'm proud of our team, is that not only are we looking at bringing precision into an area like immunology, but that also means that we're really building a company a different way. When we say things like a precision first approach or precision first company, what we're also talking about is changing the perspective of how therapies can be developed, really putting the patient and patient data first and developing therapies really with the idea of maximizing as much clinical benefit for patients across a broad area of diseases. And that leads to really designing a company a different way, running our development a different way. It's a really innovative way of looking at developing new therapies for patients, and one that we're very proud of. It's kind of flipping the traditional, molecule first approach from pharma on its head, and really starting with patient data and patient outcomes first, and then backing out to what is the best thing to use for each patient. And we've got a fantastic team doing that. So just wanted to give a shout out to them and also talk a bit about our different approach there. And I really appreciate the time that you've given us on your podcast.
And that's a wrap, as they say. Not the kind that you would get in a restaurant, but the end of the 10th podcast. I do have some interviews ready for next week, but it's not exactly taking shape the way that I want it to, but I've got a whole week to figure that out. Next week, hopefully, we will also have an announcement about a new segment for the podcast, so I'll leave you hanging with that and get started on next week's show. So, I hope that wherever in the world you are, that you have a great week ahead, and that you will join us again next time for another Beyond Biotech.